Welcome to the Scientific Sense podcast, where we explore emerging ideas from science, policy, economics, and technology. My name is Gil Epen. We talk with world's leading academics and experts about their recent research or general areas of topical interest. Scientific Sense is an unstructured conversation with no agenda or preparation. We cover a wide variety of domains where new discoveries are made and new technologies are developed on a daily basis. We are most interested in how new ideas affect society and help educate the world how to pursue a rewarding and enjoyable life rooted in science, logic, and information. We seek knowledge without boundaries or constraints and provide unedited content of conversations with researchers and leaders who love what they do. A companion blog to this podcast can be found at scientificsense.com, and this podcast is available on over a dozen platforms and directly at scientificsense.net. If you have suggestions for topics, guests, and other ideas, please send them to info@scientificsense.com, and I can be reached at gil@epen.info. My guest today is Professor William Renthal, who is Director of Research at the John Graham Headache Center at Brigham and Women's Hospital and an Assistant Professor of Neurology at Harvard Medical School. His research focuses on the use of molecular genetics to develop therapeutic strategies for headache and pain. Welcome, Will.
Thank you so much. It's great to be here.
Yeah, thanks for doing this. I want to start with one of your papers from 2019, Migraine-associated gene expression in cell types of the central and peripheral nervous system, in which you say genome-wide association studies have implicated dozens of genes with migraine susceptibility, but it remains unclear in which nervous system cell types these genes are expressed. Migraine and pain, headache, this is a very complex disease, isn't it? One of the reasons, obviously, is pain is a subjective experience. It's difficult to measure. And so you have a study here. You want to talk a bit about the study?
Yeah, so first of all, thank you so much for having me. I think this is a really important topic, and I'm really excited to share our work with your community. One of the biggest challenges that we face with, in particular, migraine, but really this could be extended to any pain disorder, is where is this problem in your body? And if you think about the nervous system, I mean, this and you may have seen these pictures of the nervous system starting with your brain, and extends these long axons down your spinal cord, and then you see these branches come off your spinal cord and go off into your fingers. And I mean, really, where in this pathway does pain exist, and where and in particular, and I'm interested in head pain, where does that exist? And it's a relatively similar problem for both migraine and pain in general. And when we think about migraine, one of the things that's really stood out for decades now is that this is a highly genetic disorder. There are cases where we have familial causes of migraine, where there's really one or maybe a few genes that are passed down from family member to family member that cause this predisposition. But it turns out in most people, it's actually what we call a polygenic disorder, meaning many genes are affected and many genes are associated with the disease. And each one of these genes, or variants, as they're called, adds a little bit of risk. And it's really hard to know exactly where that risk is coming from when you have a condition like migraine that could potentially exist in many different places in your body. And so one of the things that my research is focused on is trying to figure out where in this nervous system migraine actually comes from. And so we take large samples of patients who have either self-reported or physicians have diagnosed them with migraine, and look for variants that are more associated with migraine conditions compared to controls. And then, and what other groups have identified, and there's a large group called the International Headache Genetics Consortium, which has published some of the real seminal work in this space, have identified over 100 variants that are significantly associated with migraine. And I'm sorry, go ahead.
No, I just, it is fascinating. So yeah, when we think about migraine, we think about headache. But more generally, what you're saying is it's polygenic. We don't really know the origin of the problem, and the pain could be starting somewhere and then migrating to some other place or something along those lines.
Right. So when you think about it, is the susceptibility when a migraine attack occurs caused because of the peripheral nervous system? Basically, these pain fibers you think of when you slice your finger, it sends signals all the way up your arm to your spinal cord to your brain. Is it a problem with those kind of peripheral nerves that are in your face and head and around your brain? Or is it something that's happening inside your brainstem or brain itself? And with migraine, we really don't know the answer to that question. And it's a very complicated problem. Then the answer may be, in some people it's one way, and some other people it's another place, or everybody has kind of a different mix of all of these relative components. And so that's one of the major outstanding questions in the field. And we're hoping that the human genetic information that we now are able to accumulate across hundreds of thousands of, if even millions of people, will guide us and tell us where to look, or at least in the majority of patients. And
so, so would you call it sort of migraine spectrum then, Will? So how is the diagnosis done today?
Well, the diagnosis is actually quite old-fashioned in that I might get in trouble for saying that, but it is decades old, and it's really entirely based on symptoms. And so if people have severe headache that oftentimes is on one side of their head, although can be at both, and this throbbing, pulsating sensation, and it's associated with other neurologic deficits such as weakness or numbness, or nausea or vomiting, or light sensitivity, or noise sensitivity, or sometimes sensitivity to smell, or really tenderness to the touch of your scalp. These symptoms are all added together to give you a condition we call migraine. And so you can imagine there's a lot of heterogeneity in people who might have those same symptoms. In fact, some people who have a head injury could report very similar symptoms and don't have migraine at all. People who have infection, viral encephalitis, sometimes experience a very similar type of head pain. But it's not this episodic, recurrent condition that upwards of nearly 20% of the population, at least in women, have. So it's clearly a different biology, but maybe that's triggering these events. But perhaps the downstream function of it is really quite similar between different causes.
Yes. I have seen it run in families. So obviously there is a strong genetic component. What's the incidence rate approximately?
So it's a nearly, it's depending on the study, it can be up nearly 20% of the population. Usually high teens is what's cited. And of them, about somewhere around 1% to 2% of the population are what we call chronic migraine patients who have more than 15 migraine days a month. And that's a very disabling condition. It has a huge toll on society, both in terms of the lost productivity to the economy, but also in terms of these people's lives. I mean, it really can be dramatically affected when half of the days of your life are spent in severe pain.
1 in 5 is really, really, really high, so the percentage of that population seeking treatment, I would imagine, is a lot smaller, right?
That's right. I'd say it's hard to know exactly the numbers. It depends on the regions and some cultural differences as well. But a lot of patients are having what we call low frequency migraine. So they can take a medicine. There are a variety of them that exist now, ranging from medicines like Advil or ibuprofen, all the way to some very new medications, which these CGRP antagonists. These medications were all very effective in treating these individual attacks, and different patients need a different degree of intervention. The patients that I think are most disabled, there is this kind of 1 to 2% of the population that have these migraine attacks very, very frequently. And so those are the ones that typically show up to a tertiary headache care center, and often we try many, many different treatments for them.
Yeah.
And this is what I was just going to say. This is, for me, one of the huge goals of my research is to try to not only identify better therapeutics, but also figure out right now, when I see 10 or 20 people in a day, which patients should be on which treatment. And the way we currently make these decisions are by asking people which side effects they prefer the best. And that's, if you think about it, very, very archaic in terms of our approach, given the fact we can now sequence your entire genome in a day and tell you which cancer therapeutics based on the sequence of your tumor. And then, that's just happening in the hall across the way from me. And for me, I'm asking people, would you rather be asleep all day, or would you rather not be able to find words in the morning? These are, this is how I make my decisions, and I feel very behind the times. So, one of the things we're really trying to do is figure out, is there any way we can understand a treatment response in patients, based on their genetic information or other biomarkers.
Yeah. Just very, very quickly, regular painkillers, do they help here? Do people self-treat? I'm just going back to the 20% incidence rate. That seems really high.
Yeah, most people try many treatments before coming to see a physician, usually over-the-counter medicines like ibuprofen or Excedrin, which includes a little bit of caffeine, are the typical first-line treatments that people will try, and oftentimes are effective to some degree. It just depends on the severity of the migraine attacks, and oftentimes this will change throughout an individual's life. And so at that point, sometimes people will need to seek care at one stage in their life, and otherwise are completely fine.
Was there any connection between this and the opioid issue we ran into?
So there is a relationship. It turns out that for a variety of reasons, opioids have been tried for treating migraine, and quite frankly, they're not that effective. But I think a lot of physicians who didn't really keep up with the headache literature would try it just because people were in pain, and it certainly does make you feel better in the short term. The challenge is that chronic opioid use, especially for headache, isn't very effective for long term, and can actually increase the likelihood of having more severe headaches down the road. It's a condition that we kind of term medication overuse headache, which has really classically been associated with chronic opioid or barbiturate use. Some people have argued that perhaps this condition exists for other pain relievers as well, such as ibuprofen. I think the data for that is a little bit less strong, but certainly for opioids, there's a pretty good consensus that this can actually worsen your headache if you use this regularly.
And so going back to the paper, so you're looking at 54 migraine-associated genes, and they're all over the place. So what did you find from the study?
Yeah, and so maybe just taking a couple of steps back, what we were out looking for was is there any? I'll just restate this. There's a major debate that we have at all of our national conferences and international conferences, and that is migraine in the central nervous system or the peripheral nervous system? And there are some different camps in our field, and people are constantly arguing and providing support for one versus the other. And I think right now there's reasonable good support that both are involved, and no one knows where it starts. So we were thinking that if we could take people's genetic information, maybe we could learn something about what's really the proximal trigger for this. And it turns out that when we look at the genes that are associated with migraine, they are, as you said, kind of all over the place. They're expressed in blood cells, and they're expressed in peripheral neurons that are accessing central neurons. And so the answer to that question did not solve this age-old debate, but perhaps provided some support that that maybe both camps are right. And I think that maybe different individuals have susceptibility to migraine because different parts of this pathway are affected. So maybe some people have neurons in the central nervous system that are hyperactive that increase their likelihood for triggering a migraine attack. And other people may not have anything wrong with their excitability of their central nervous system, but their pain neurons in their face and around their head are just really easy to get kind of stimulated. They get activated at a much lower threshold than someone else, and so they might experience the pain associated with migraine much more easily. And so I think that's the way I'm starting to conceptualize this, and the next real step is to try to understand how these individual variants that have been associated with migraine actually cause dysfunction in the cell types in which they're expressed.
Yeah, would it be correct, Will, to think about migraine as sort of a diverse set of diseases that all have some sort of symptoms? So we're using symptoms as a diagnosis, but as you say, it could originate from many different parts of the body, right? So are we calling migraine sort of a bucket of different diseases?
I think that almost certainly there are different categories of migraine within this kind of broad stroke diagnosis for sure. The question is, what is the most meaningful way to separate people out? And right now, if you respond to the medications we use to treat migraine, such as sumatriptan or some of these new CGRP antagonists, we're comfortable with calling that migraine. But for people who don't respond, there's a real value to separating people out to the medications that they are most likely to respond to. And I think that's kind of the next step here, is to figure out who's going to respond to which medication first. And I should just kind of take a step back and say, when people have chronic migraine, meaning more than 15 days of this a month, we often have to start out with one medication and then try another and then try another. And this can go on for months or even years of trial and error of different medications. And I think that the real value would be if we could predict, based on what type of migraine a person has, which medication they're most likely to get benefit from. That's where the real value comes in separating people based on the subtype of their migraine.
Yeah. You have another study, erenumab, which is a therapeutic used for treatment of migraine, CGRP receptor monoclonal antibody. You say it's for preventative treatment of migraine, but it has some severe adverse effects. You want to talk a bit about, and you're using some way to predict who might have an adverse event?
Right. So one of the things that's been a huge breakthrough in our field in headache and in migraine medicine over the past few years has been the development of these new medications that target a molecule called CGRP. And CGRP is a peptide, or calcitonin gene-related peptide, that is secreted by some of the pain neurons in your head and around in your body as well. And this peptide can create kind of an inflammatory response, so to speak, inside your blood vessels and around your head, and it can also signal to other neurons in your head, in your brain. And what's been found is that when people have a migraine attack, this peptide gets released more frequently. And if you inhibit this peptide, it reduces the severity of migraine-related pain. And so this work actually was recently just awarded the Brain Prize, which is a really exciting thing for the field. And the investigators who made these major seminal discoveries received this. And we now have drugs to treat, to block this pathway, both monoclonal antibodies that just sequester all of the CGRP in your blood, or monoclonal antibodies that bind to the receptor and prevent it from being activated. And then now there's even more recently small molecule antagonists that block the receptors directly. And so this new kind of this new tool in our toolbox has actually been really effective for a lot of patients. But one of the complications is that the CGRP peptide is not unique for the pain system. This is also used, as evolution tends to do, repurposes molecules for different systems in your body. And one of the questions we asked was, can you use some of these new technologies that are available, by that I refer to single cell genomics, to predict where the CGRP peptides are likely to be acting, and therefore might be able to predict some of the off-target effects. And so we went through some databases that we and others have generated and found that perhaps the part of the body that tends to express CGRP the highest, at least in the data sets that we have, are actually enteric neurons, meaning that these are neurons that promote peristalsis in your gut, and you need to help kind of digest your food. And this led us, I think, in early 2018 to propose that that maybe constipation is going to be a major problem with these medications. Surprisingly, the clinical trials largely, well, there was a small signal on a couple of them. The major clinical trial for erenumab, which is one of these CGRP monoclonal antibodies, didn't really show a statistically significant signal for constipation in these trials. And so we were surprised by that, as I think many of us still are, that didn't show up. And given our experience in the real world now, I mean, this is one of the most common complaints by patients who are taking these medications. I should just say they're rarely limiting in treatment. Sometimes they are, but most people are able to take take over-the-counter supplements to prevent this from being a major problem. But in rare cases, it can cause complete ileuses and or places where you cannot pass stool, and that's a real medical emergency. So this has not been something that has really prevented the use of these medications, and it's very, very rare. But it is something that we didn't really detect during the clinical trials, and something that's kind of come out afterwards. And so I think the real point of that study was just to highlight that many of these things can be predicted ahead of time by some of these new single-cell genomic technologies that we use.
And so practically, Bill, how will it work? So you'll take a blood test or something, and it will tell you some sort of probability that you might have a serious side effect like constipation.
We actually can't yet predict who's going to have the side effect and who isn't. What we're actually proposing in that study was that these data sets that have been generated both in animal models and also now more increasingly in human tissues, we can use these data sets that are being generated to predict where very targeted therapies will act. So it's more of use, I think, in a very early stage of new therapy developments to think about what kind of side effects to test for. And I think this is something that will increasingly become standard both at pharmaceutical companies and also perhaps by the FDA in requiring people to think about potential toxicology and other off-target effects.
Yeah, so I guess there's some reporting bias in the clinical trials. So if you have some expectation of certain type of side effects during trial, you can look for them.
Exactly. Well, I mean, I think one part of it is the bias from the patient, but in terms of placebo responses as well. But I think from the standpoint of just understanding the safety of a new molecule, I think, and especially as we become more and more targeted with these therapies, I think these data sets will allow us to provide safer treatments, or at least more rational characterizations of these in clinical trials.
Yeah. I want to finish up with your most recent paper, Transcriptional Reprogramming of Distinct Peripheral Sensory Neuron Subtypes After Axonal Injury. So the injury to axons result in migraine type issues?
Not exactly. So this work is a little bit tangential to my work in migraine. The reason why I'm working in this space is because, well, for two reasons. One is because axonal injury is a very common cause of pain. So you think about postoperative pain or post-traumatic pain. This is largely driven by axonal injury or inflammation in a particular area. And so when we're thinking about developing new pain therapeutics, for us it was a very important model to use to try to identify new pain targets. And so when we think about pain targets that work peripherally after, let's say, an injury to your leg, there's a chance if that molecule is also expressed in the neurons in your head. So this is the trigeminal system, as we call it. The new pain target might actually work for both peripheral and head pain. So that's kind of the link between the two areas of my research. And so really I'm focusing on trying to understand new pain targets.
So Will, what exactly are axons?
So axons are the parts of a neuron that transmit the action potential. So when you hear about neurons firing, so to speak, what's happening is actually there's an electrical signal that's being carried from one part of the cell down a long shaft, so to speak, that connects very near another cell, and that's how the information is processed. There's this electrical signal that goes down the axon, results in the release of neurotransmitters at the end of that axon terminal that then get perceived by the neighboring cell, and that's how the electricity, so to speak, is communicated from one place in the brain or peripheral nervous system to another.
And I might not have understood this well. So how do they get injured?
Well, when you think about anytime you hit your finger with a hammer or cut yourself with a knife, the axons that are part of your are buried inside of your skin that detect all these light touch or temperature or cold or any of these senses that you have. When you damage your tissue, the very ends of those axons actually get damaged themselves. And so that injury sends a bunch of molecular signals back to your, first to your spinal cord and then to your brain, that there's been damage that occurs, and that lets you to know to protect that limb or finger or whatever it is that got injured from the environment so that it can heal. And so there's an obvious, very important evolutionary need for pain. But in some cases, which is really where I come in, from a clinical standpoint, the pain is carried on for a long period of time after any type of injury. Or in head pain, there's really no injury. It's just the system's getting activated pathologically. And so there's a huge amount of research in my group and in other places as well is really aimed at trying to understand how to turn these kind of crazy neurons off so that in patients who don't have an active injury going on, we can help them have relief and live normal lives.
So some sort of trauma to some part of the body results in some sort of injury in the axonal level, and even after the body has healed, if I understand this correctly, Will, the neurons are sort of still firing, and you still have pain.
So it depends. So this is a kind of an important area in the field. For instance, what we see in many patients, they'll go in for some surgical procedure, either a knee repair or gallbladder removal, something like that. In most people, this involves an incision, and that incision will occur, and they'll be sewn up by the surgeon, and then a few weeks later, there's no pain, all the inflammation is gone, and everyone's back to normal. But in a small set of patients, for some reason, and we don't understand the mechanisms of this, those neurons do not, even though the skin heals up and everything looks normal, those neurons don't go back to normal. They continue to fire, or they send signals into the central nervous system that makes it perceive the signals differently. This is a process that is called central sensitization, where you become sensitized to otherwise innocuous stimuli when you didn't, just because of some kind of rewiring that goes on, or not so much rewiring, but kind of different thresholds are set in the way a neuron fires that makes it fire more easily the next time it gets activated. And so this problem is something that we're trying to understand. And if we could understand that, I think that would significantly help situations like this opioid crisis that's going on, where we're having to give these patients really high doses of medication to provide a functional life.
Yeah. Does it have to be a sort of a severe trauma for this to happen?
It does not need to be severe, and that's another kind of mysterious thing here. I mean, I guess it depends on what you mean by severe, but I mean, you don't have to be in a major car accident. It does require injury to the limb or to the head or whatever system you're talking about, but it, a major injury.
And so going back to the paper, so transcription, you say our findings suggest that transcription factors induced early after peripheral nerve injury confer the cellular plasticity required for sensory neurons to transform into a regenerative state. So could you explain that a bit? So what are transcription factors?
Transcription factors are proteins that are in the nucleus of a cell that tell the cell when to turn on and how much to turn on a given gene. And so they're kind of the master regulators of gene expression, and those genes are what kind of make the RNA, which then makes the proteins, which are the building blocks of your cell. And so they kind of they set the thresholds and the expression levels for all of these genes and proteins. One of the things that we really wanted to understand was we made this kind of bizarre observation that after sensory neurons are damaged, their gene expression profiles dramatically change. And it turns out that these genes that are turned on are important to regenerate the axon that got damaged. And so we spent a lot of time trying to understand this process in exquisite detail. And within different cell types within the sensory neurons, there are many different types. Some detect touch, some detect cold, some detect heat. And so we were able to map out the responses in each of these cell types. They turn out to be remarkably similar, but it's still, from a therapeutic standpoint, that actually might be really useful. And we think that in some patients, especially if you think about it, like phantom limb pain, where a leg is amputated, the nerves aren't able to regenerate properly because they don't have their end organ to grow back into, and so we think that by better understanding these mechanisms that drive regeneration of the axon, we can, in some cases, actually prevent these cells from being as hyperactive and angry, so to speak, after surgical procedures.
Yeah, I mean, it's a bit like, I hate to devalue it, but just like an electronic equipment, given the wrong voltage, stays on even after you turn it off. It seems like the brain is on. You just have to sort of reset it somehow, right?
We try to do just that. In many of these patients, unfortunately, unlike in electronics, we can't turn it off for a very long period of time, but we do actually administer anesthetics to the peripheral nerve to try to essentially, we do just like you would with your phone if it's acting weird. You turn it off and then turn it back on and see if it gets back to normal. And that's exactly the basis of nerve blocks, where you'll go to the doctor and they'll inject the anesthetic into your leg and hope that the nerve comes back normal. It does relieve the pain, but oftentimes it comes back after days, weeks, months, and still at a very high level. So one of the major outstanding kind of holy grails of pain medicine is to develop tools that will allow us to keep these neurons quiet for longer periods of time without causing complete anesthesia, right? Because if you imagine lidocaine in your leg, you can't feel anything that that's been touched. Or when the dentist puts Novocaine in your tooth, you can't even bite your tongue. It's dangerous to not be able to feel. And so what we and many other groups are trying to do is develop pain medicines that will turn these nerves off, but not lose the sensation of touch so that you can safely anesthetize these pain neurons without affecting the other ones.
I don't know what the right term would be, but this sort of perpetual neuronal activation, in this case, because of a physical injury, do you think things like this happen in more, psychological things like PTSD?
It's a different mechanism. So in what would be kind of set, so there's a couple of other, I think your question has a few questions buried in it. With the perception of pain, obviously the perception of pain occurs in your brain. This is not something that is driven just by the sensory neurons in your hand. In fact, there are patients who have strokes that are in the brain, don't even involve the sensory neurons in their hands and feet and face, who have chronic, really debilitating pain because it affects the pain pathways in your brain. And so there's clearly neural circuitry in your brain that mediates the sensation of pain. And so in more psychological, or what's classically called psychological conditions like PTSD or even depression, where there's very high comorbidity with heightened pain or reports of pain, there may very well be circuit level changes that occur in these pain circuits in your brain that make you more sensitive to the perception of pain, even though the stimulus was otherwise the same as it was, let's say, before that condition developed. And so I think absolutely there's an overlap, but perhaps at a different level than what I'm talking about with the axons that get injured by a trauma.
Yeah, I mean, it would be fascinating if there's some sort of therapeutic approaches are overlapping between the two.
I think with this more central, these circuitry involved in pain that's in your brain, there are a large number of investigators that are very interested in transcranial magnetic stimulation, where they can actually activate various regions of your brain with a magnet and try to help reduce the sensation of those pains. And I think that's work that's really kind of in the beginning days. And I think as those tools become more targeted and more powerful, we'll be able to modify some of these circuits directly. We certainly can in animals. There's really great work that was done by a new investigator at Penn named Greg Corder, where he's able to control how an animal perceives pain without actually, by turning on or off certain neurons in the brain. And it's actually not just perceiving pain, but just how much they care about the pain. It's really interesting work where they can turn those cells off and give the same mouse the same stimulus, and it doesn't care about the pain. And then a few minutes later, turn those neurons back on, and the animal is very affected by the pain. And so I think there's really exciting work at the basic animal level going on in that area right now, but we're still a little ways away before clinical translation of that work.
Yeah. So in conclusion, Will, I know that your lab is doing a lot of work in this area. So if you look forward 5-10 years, where do you think we are most likely to make significant discoveries and improvements?
With regard to migraine, I think there's some really exciting new work that's helping us understand that migraine likely begins in the hypothalamus, which is an important kind of hormonal regulatory part of your brain. And I think that there's some really exciting work that's coming out looking at the role of hypothalamus and perhaps some of the hormones that are regulated by the hypothalamus in migraine. I also think that there are a variety of there's a large number of studies that are coming out with regard to the genetics of migraine that I think will help point us towards new directions that have been previously maybe overlooked or not known about. And I think from the perspective of pain, there's the new single cell genomic technologies that we have that are kind of enabled us to, for the first time, learn what molecules are expressed in the neurons that transmit pain versus other neurons, will really lead to an explosion of new tools and potentially therapeutics that will allow us to very selectively modify these cells without affecting other senses. And I think that I would look for those things in a few years, and we can touch back again then, and hopefully we'll have at least a few of them. So.
Yeah, I mean that is the area, right? So sort of understanding at the molecular level what happens. I think we are getting maybe more and more information of the genetics aspect of it, but how the machine works mechanistically is still not
I think
well
bridging
understood.
those gaps. I mean, I think because we cannot go inside people's brains on an individual level and assay specific neurons, I think we really are limited to the things we can ask people about with words, with imaging of their brains during various tasks and their genes. And those are the things we can actually directly test in a given individual. And so we have to use work in other organisms to piece those pieces together. So that's where we are. And I think there's a lot of really exciting work going on now, thanks to advances in optogenetics that allow us to turn certain neurons on and off very rapidly. And I think we'll be able to understand these circuits. It's just a matter of time.
Right. Excellent. Yeah, this has been
Absolutely.
great, Will. Thanks so much for spending time with me.
Thank you.
Thank you.
This is a Scientific Sense podcast providing unscripted conversations with leading academics and researchers on a variety of topics. If you'd like to sponsor this podcast, please reach out to info@scientificsense.com.